Hi children, the fifth video will be wrapping up with this chapter and we are dealing with the steps. It's a rewind of all the steps in the right sequence. Okay, so once again this video will be dealing with the different steps in the correct order of creation of rDNA. So the steps in a schematic representation, the steps involved in the construction of rDNA as earlier I have given and as given in your textbook. A foreign DNA is isolated from the source organism, a vector DNA is also isolated, then we use a restriction enzyme to cut the foreign DNA as well as the vector DNA, and finally we get the vector DNA with an open space, and into that this small sequence of gene of interest is inserted. Now this becomes an rDNA, and finally this rDNA is incorporated into the host for multiplication. Okay, and this we know was accomplished by Stanley and Cohen. Isolation of foreign DNA from the source organism is the first step. The source organism can be a plant or bacteria, fungi, or even an animal cell. The second step, cutting of the foreign DNA as well as the vector DNA at specific sites. Then the third step is gel electrophoresis. The purpose of gel electrophoresis is to separate the DNA fragments because they will be of different sizes. Fourth step is PCR, to make multiple copies of the foreign gene or the gene of interest in vitro. Fifth, ligation, ligating the foreign gene to the plasmid using ligase. So we get an rDNA. Next we have to insert it into the host, but the host cannot take it up. So the host competence forms the sixth step. Then last, this host with the rDNA. Now that host is called a transformant. It is put in a bioreactor. The purpose of the bioreactor, it provides optimal conditions for the multiplication of the E. coli, which is the commonly used host. And last is the downstream processing, which is involving a series of processes during which the product has to undergo changes to be finally marketed. Now among these steps we have not gone into the elaboration of the first step, third step, fourth step, seventh, and the last, downstream processing. So in this video I will be elaborating these steps. One, that is the isolation of foreign DNA from the source organisms. Then the third step, gel electrophoresis. Fourth step, PCR. Seventh step, bioreactor. And the last step, downstream processing. The intervening steps were already dealt with when I took the different tools of genetic engineering. So we are going into this sequence. So the slides that are going to be presented is all in the correct sequence. We are maintaining the same sequence, the correct sequence, all 8 steps, but already the ones which I have taken, I will just give a brief view. But the ones which are new will be elaborately discussed in this video. So isolation of DNA, the very first step which was not taken, or now only it comes, only in this part of the text it comes. How the DNA is isolated. When we learn that outline of the construction of rDNA, we just simply mentioned that DNA is taken from, foreign DNA is taken from the host and plasmid is taken from an E. coli or from a bacterium. Now how is this isolation of DNA occurring, or how is the procedure? So first of all we must know that this DNA, it is inside a membrane. It can be the membrane or the cell wall. So let this be the cell wall. And this DNA is inside the nucleus or not inside the nucleus, maybe in the nuclear if it is in the case of bacterium. So whatever it is, the first obstacle or the first hurdle to be removed is the structure that encloses the DNA. Okay, immediate surrounding is the nuclear membrane, but the nuclear membrane has pores, so that is not an issue. So I am erasing the nuclear membrane and only the biggest hurdle is the cell wall. If it is a plant cell, so now here you have to indicate, if it is a plant cell, the cell wall can be removed with the enzyme cellulase. So which plant cell? It is cellulase. We know why cellulase, because the cell wall in plant cell is made up of cellulose. If it is fungus, you know the cell wall of fungus is made up of chitins. So what is the enzyme used? Chitinase. So we can treat with chitinase and the cell wall gets ruptured or destroyed. And if it is bacterium, we know it is having a cell envelope and it can be removed with the help of the enzyme lysozyme. Okay. So first of all, the first hurdle that should be removed is the cell wall. So the cell wall is removed. So how is the cell wall removed? That depends on the source organism. If it is plant cell, we treat it with cellulase. So the cell wall is destroyed. If it is fungus, treated with chitinase, the cell wall gets destroyed. Or if it's a bacterium, you treat it with lysozyme, the cell wall gets destroyed. So now we have only the DNA, but this DNA is not in the pure form. It is associated with proteins. We know non-histone proteins, histone chromosome proteins and histone proteins which holds the DNA. We have learned packaging of DNA. So it's associated with proteins, it's associated with RNA, it is also associated with some lipids. I'll show with green. So the DNA is not in pure form. It is associated with proteins, RNA and lipids. So we have to get rid of these structures so that we can get the DNA in its pure form. So to get rid of proteins, we have to use the enzyme protease. So treat it with protease. So I am going to remove all the proteins. Okay, the blue color was the proteins. So all the proteins were removed. Then I have to get rid of the RNA. So I must treat it with RNases. So RNA is got rid. Then I must treat with, to get rid of the lipids, I have to treat it with lipase. Now, after treating with lipases, what happened? Even lipids are got rid. Now we got DNA in its pure form. Okay, now DNA is in the pure form. So once again, the DNA is enclosed in a cell wall. The cell wall should be removed. For removing the cell wall, it depends on the source organism. If it's plant, treat it with cellulase. If it's fungus, treat with chitinase, and if it's bacterium, treat with lysozyme. So what happens as a result? The cell wall gets destroyed and removed. Then this DNA is not in the pure form. It is associated with RNA, it's associated with lipids and also with the proteins. We know packaging, the proteins like histones and non-histone chromosome proteins. So we have to get rid of all these with the respective enzymes. To get rid of proteins, make use of protease. So all the proteins are removed. Then to get rid of RNA, treat with RNases. All the RNAs are removed and lipids with the help of lipase. So after removing all that, what happens? We get the DNA in its pure form. Now this DNA in a centrifuge tube, it is always done in a centrifuge tube. It is subjected to centrifugation and it is down, there are the debris, that is that destroyed cell wall, destroyed proteins, destroyed RNA proteins all settle as debris at the base of the centrifuge, and in the supernatant we have the DNA, but we have to precipitate the DNA out. For that we have to add chilled ethanol, and this should be added through the sides. When it is added through the sides, what happens? This DNA gets precipitated to the top and it can be easily spooled out with the help of a glass rod. So we have the DNA in the pure form, but it is at the base of the centrifuge tube. So to precipitate the DNA, what we have to add? We have to add pour chilled ethanol through the sides. After pouring chilled ethanol, what happens? The DNA rise, and with the help of a glass rod we can spool it. Okay, so now these are the steps. The first step is indicated here. In this step we are destroying the cell wall. So it depends on the source organism. If it's plant cell, we are treating with cellulase. If it's Fungus we are treating with chitinase, of its bacterium we are treating with lysozyme. So in the first stage the cell wall is destroyed. In the second stage we are destroying the proteins, RNA and lipids with the respective enzyme. Proteases to destroy proteins, RNases to destroy RNA, lipases to destroy all the lipids. So we have the DNA. Now we have the DNA in the pure form. But this DNA is present in the supernatant and the debris. This is indicating the debris. All the ruptured structures are settled. Now we need the DNA to be precipitated. We need the DNA to get precipitated. So this is very important. We have to pour chilled ethanol. See, through the sides we pour chilled ethanol. When we pour chilled ethanol, what happens? The DNA rises, and with the help of a glass rod, you can take it out. This process is called spooling. That is taking the DNA from the precipitate which has precipitated in chilled ethanol with the help of a glass rod is called spooling. This procedure we will be doing in the lab. Isolation of DNA from banana or pumpkin or coconut kernel. We will be doing all this in the lab. So I hope you got a clarity of how DNA is isolated. Clear? Next step we have already dealt with. After getting the DNA, we know our gene of interest and we make use of restriction enzymes. See, this is the restriction enzyme. Okay, and definitely this is a restriction enzyme EcoR1. That's why it is cutting. Its recognition sequence is GAATTC, and it cuts between G and A. So it cuts between G and A, and we know what are the ends. Describing the ends: overhanging, single-stranded, sticky ends. And we know why it is sticky, because it forms hydrogen bonds with its cut counterparts. All this I have already dealt with in the previous slide. Now what is new here is the restriction sites here displayed is just one restriction site. It is not always one restriction site. There may be many restriction sites. It is always better to have, they say when we learn the features of a cloning vector, it is always better to have single restriction site. Okay, that is in the case of a cloning vector, but in the case of a lengthy 2.2 meter long DNA of ours, there are chances of many restriction sites, that is GAATTC, there is chance of that to get repeated. So I am going to extend this. So I have extended this. So here we have understood that's one restriction site. Restriction site, cloning site or recognition site. It is that recognition sequence, and this is for EcoR1. The recognition sequence is GAATTC and it cuts between G and A. So here in the diagram as depicted, there's one restriction site. Now you check, I have extended this. Here there's another restriction site, and here you check, here there's another restriction site, and here also there's another restriction site. To check whether this is correct, just see the following sequence. See the sequence is GAATTC. Look at the sequence here, GAATTC. Here GAATTC and here GAATTC. So how many recognition sites or cloning sites are there? 1, 2, 3, 4. So how many segments will we get after the restriction enzyme or restriction endonuclease cuts the strand? We will get how many sequence? Just draw 1, then 2, then 3, then 4, 5. Now look at the sequence. If I am highlighting it or making it broad, look at the length of these segments cut by the restriction enzyme EcoR1. Look at, there are many fragments of different size. So what will be the next step? Common sense, it warrants the step which sorts these fragments according to the size. So the next step that is mandatory after cutting with restriction enzyme is gel electrophoresis. So this is the step that is going to be elaborated in the next slide. It's something new. we have learnt already in DNA fingerprinting, but now we are going to learn in detail. Why gel electrophoresis? The reason is that restriction enzyme cuts at restriction sites, and restriction sites are not one of few, there are many restriction sites. As a result, wherever there is recognition sequence, this restriction enzyme cuts and we get a number of fragments of different size. So what is necessary is to go for gel electrophoresis. Gel electrophoresis is an agarose gel slab. See, this is an agarose gel slab in the form of a slab. It is made up of a seaweed, agar, and electricity is made to pass. See, electric current is made to pass, and here you can see some wells. Wells means some incisions. Into these incisions, cut fragments are fed or put. Okay, and then current is made to pass. The basic principle is that our DNA, being negatively charged, gets forced to move towards the anode because it's negatively charged. Okay, so when an electric current is passed, it moves towards the anode and it gets sorted according to the size. The bigger ones are close to the well, whereas the smallest one move further apart. Okay, so this is going to be the smallest fragment. Okay, so due to the sieving effect caused under the electric field of gel electrophoresis, the DNA fragments get arranged according to their size. Okay, so now we got it arranged according to their size. Now, you should remember, when we do it in the lab also, when we isolate the DNA, you can see the color of the DNA. It's off-white in color. It's not milky white, it's off-white in color, and the gel is also off-white. So this DNA blends, or it is camouflaged in this gel. It's not visible to the naked eye because it matches with the gel. Both are off-white in color, like candle wax color. Okay, so to identify this DNA, we have to add a dye, and this dye is ethidium bromide. And immediately after adding ethidium bromide, still it's not visible to our eye. You have to expose it to the ultraviolet rays, to sun's rays. And what happens that time? It will appear to be bright orange bands. So this picture is depicting an X-ray view of this banding pattern. Okay, an X-ray of this is taken to show that the smallest moves further away. See, the smallest band moves further away. This is the largest band towards the wells. These are the wells: well 1, well 2, well 3, well 4, well 5. Okay, there are the wells and the banding. Okay, the smallest one moves further away. This is just an X-ray of this. Now into this, I told you, no, this is not visible to our naked eye because it matches the DNA fragments matches with the background of the gel. So to stand out, we have to add ethidium bromide. So in this ethidium bromide, remember the stain, it is, or dye, ethidium bromide is added and immediately exposed to ultraviolet rays. Now you can see the banding. It appears as bright orange bands. See, you can see the bright orange bands, fluorescent orange. Okay, then the next step is very, very important. Next step is elution. Elution is, we need not take out all the DNA fragments that are separated, only with the fragment which has our gene of interest. For example, this is the fragment. Take another color which stands out. Let's assume this as the fragment of DNA which has our gene of interest. So only that is cut out. See, look at this picture where that particular fragment, it's cut out or extracted from the gel. Look at the gel, it's cut out. Only where there's the gene of interest, it's cut out. And the gene of interest will be only one. Okay, maybe the gene encoding for insulin or gene encoding for coagulation, etc. Okay, so that is only being cut. So once again, purpose of gel electrophoresis is because of the restriction enzyme. It cuts at the restriction sites, and the restriction sites are many, and therefore what is the result of cutting? It generates a number of fragments of different size. So this is required. Gel electrophoresis arrange or separates the fragments of DNA that are cut by the restriction enzyme. This comprises an agarose gel. Electricity is made to pass, and under this electric field, the DNA being negatively charged is forced to move towards the anode. And after due to the sieving effect, we can see the DNA fragments gets arranged according to the size. We can see the smallest one moves further away. Okay, and this is not visible to our naked eye. Immediately from that step is this step. That is, to make it visible, we add the stain ethidium bromide and then expose it to ultraviolet rays. So, if this is the first point, the second point is this, then it is stained with ethidium bromide and then exposed to ultraviolet rays, and the bands appear to be bright orange color. Then what? The third step is elution. Only the strand or the fragment of DNA which has our gene of interest is cut out or extracted from the gel slab. This process is called elution. Okay. So, in 3 steps, gel electrophoresis is occurring on the agarose gel. Then you are staining with ethidium bromide and exposing to ultraviolet rays, so it stands out. The color takes up is bright orange, and finally we are cutting out that particular strand which has our gene of interest. Next, we got that particular strand having our gene of interest. Let this be our strand which is selected. We saw the last stage coming under gel electrophoresis, that is elution. After elution, we got this gene of interest. Can we go ahead with constructing of the rDNA? So, this is the plasmid. Into this plasmid we are going to put this. Can we go ahead with this process? No, we have only one gene of interest. So, if at all somewhere this further process, there is a flaw, that gene of interest is lost, and again we have to start from the first process, the isolation of DNA, then cutting, then gel electrophoresis, and then we have to make the rDNA. So, the better option to do the step at ease, make copies of this gene of interest. So, very, very important, make multiple copies of this gene of interest. This is the gene of interest. So, before going into this step, that is construction of the rDNA, make many copies of the gene of interest. So, you can do the further steps at ease. If at all there is a mistake, you have the gene of interest. You need not start from the beginning. With the gene, many gene of interest are there because of this technique called PCR, and all these gene of interest will be stored in the gene bank. Okay. So, once you make a mistake or falter in the next step, you can take with the next gene of interest which is there in the gene bank. Again mistake, you can do trial and error method. So, this is very mandatory to go for PCR, especially in the case of crime detection, parentage detection, etc. In DNA fingerprinting, we get the gene of interest only once from the person. Okay. Once we get the DNA from that person or from the crime site, it is not easily available. Maybe the person is dead, so we might take from autopsy, we will take. So, what we have to do is after we get the gene of interest, make many copies and store it in the gene bank. So, at any time we can conduct experiments further. Okay. So, this is very important in DNA fingerprinting and any type of crime investigation, because the criminal or the victim will not be available for supplying the DNA. So, the gene of interest is multiplied. So, purpose of PCR is multiplication of gene of interest or alien DNA or foreign DNA in vitro. Very important. Where it is multiplied? Outside the body. So, once again, what is PCR? PCR is a technique of multiplying the gene of interest in vitro. The full form is polymerase chain reaction. It involves 3 steps. The first step is denaturization. So, look at this DNA. All of you focus at this DNA. It is a double stranded DNA. Heat is applied, and when a lot of heat is applied, what happens to 2 strands of DNA? It separates. So, this will result in the separation of the 2 strands of DNA. One strand is running the 5' 3' direction, the other strand is running the 3' 5' direction. So, they separate. That is how, with the help of heat. So, the first step is application of heat to separate the 2 strands of DNA. That is called denaturization. Support with this diagram. Then the second step is annealing. Annealing is addition of primer at the 3' end of both the strands. So this is a primer. And this primer should be complementary to which end of the strand? At the 3' end. It should be complementary to the sequences at the 3' end. If suppose the 3' end is AAA, the primer should be definitely TTT. Here also the primer is AAA. So the DNA is TTT and so the primer is AAA. Okay. So the primers are chemically synthesized oligonucleotides complementary to the 3' end. So the primers added. Definitely they will go bind at the 3' end where it is complementary to their base. Because their base is AAA, definitely it will go to that DNA strand which is TT. Here also it goes to TT. Okay. So I hope you have understood annealing. That is simply at all it is this addition of primer. Then the third step is extension. After that primer is added, this primer should be extended. See, from here it should be extended this side, and from here it should be extended this direction. Okay. So the primer should be extended. So we have to make use of an enzyme, DNA polymerase or Taq polymerase. The function of polymerase is polymerization, adding of nucleotides. And here also remember the property of a polymerase enzyme. It functions on the 5'. It functions in the 5', 3' direction. Therefore, the strand which serves as a template will be the 3', 5' strand. So you can see in the first case, the strand is this 3', 5'. Okay, so the DNA polymerase functions in this direction. Okay, and second case, look at the parental strand. It is running the 3', 5' direction. So our DNA polymerase is adding in this direction. See the way it's the direction I'll draw with green, so it stands out. See the way the DNA polymerase is acting. In the strand, down strand which is running the 3', 5', 3', 5' direction. See how is it? It's running on the 3', 5' direction strand. And on the top strand also you can see the top strand 3', 5'. Look at the direction this way. Look at the arrowhead. Please concentrate on the arrowhead. So you must maintain the polarity of the template strand. The template strand should be running in the 3', 5' direction. Why? The DNA polymerase functions in the 5', 3' direction. As a result, you get how many DNAs? 2 daughter DNAs. And now 30 cycles. If this is repeated 30 times, you can get nearly 1 billion copies of the gene of interest. So once again a recap of the entire process. First step is denaturization. It is the application of heat to separate the 2 strands. So you are depicting it here. Heat is applied and the 2 strands get separated. The second step is annealing. Annealing is addition of primer at the 3' end. If our 3' end is CCC of the DNA, it is CCC. So what will be our primer? Our primer will be GGG. It's complementary to the 3' end of the strand. Okay, so what are these primers? These are chemically synthesized oligonucleotides complementary to the 3' end. So you are adding primer. That process called annealing. And the third step is extension. The primers are extended. Look at the way it's extended. Look at the down strand. How is it extended? In this direction. Look at the direction. It runs in the 5', 3' direction. And look at the top strand. Top strand is also maintained in the 5', 3' direction. Okay, the working of the DNA polymerase. Look at the arrow. So the template will be running the 3', 5'. So that is extension is primers are extended by DNA polymerase. Okay. And 30 cycles of this will produce nearly 1 billion copies of the gene of interest. Now the specialty of the DNA polymerase. We know enzymes are very sensitive to temperature, pH, etc. But here this particular enzyme is thermostable. It can withstand the heat. It does not get denaturized. When you do lab experiment, we see how the salivary amylase, with the help of high temperature, how we can destroy it or denaturize it. But here, this particular enzyme, Taq polymerase or DNA polymerase, is thermostable. it can withstand very high temperature. So, it does not get denaturized, it does its role of polymerization. This particular enzyme is derived from the source organism Thermus aquaticus. It is a bacterium. So, this DNA polymerase is obtained from the source called Thermus aquaticus. The speciality of this enzyme DNA polymerase is that it is thermostable. It can withstand high temperature because a lot of heat is provided to make the two strands of DNA separate. Okay. So, PCR is very, very important because it is we cannot do trial and error with the gene of interest. So, we have to make many copies of the gene of interest in vitro and store it in the gene bank, and therefore, PCR or polymerase chain reaction is very, very important. Next, we finally made a lot of copies of the gene of interest. Okay. Now, many copies of this by PCR, and it's stored in our gene bank. Now, this can be incorporated into the plasmid and we will get the rDNA. We know this stuff, we have learnt it already, and we to know about the speciality of the cut ends, single stranded, overhanging and sticky ends. Now, this rDNA is going to be incorporated into the host. Next step, this also it does not easily get into the host. It has to undergo competence because the rDNA is or DNA is hydrophilic. It cannot be easily taken up by the host. So, it is subjected to chemical treatment. First, it is treated with certain specific concentration of calcium ions. Okay. After that, both the rDNA and the host is placed on ice. Then heat shock and again back on ice. So, this is a sequence of step, rapid sequence. Ice, it is incubated, both the host and the rDNA is placed on ice, then heat shock for a brief period of 40, and the heat shock is of 42 degrees Celsius for just 30 seconds, see, just 30 seconds, and then back to ice. When this treatment occurs, rapid treatment occurs, what happens? Pores are formed in the host and the rDNA gets in. That is how the rDNA gets easily into the host. That I have already discussed in the previous videos, that is why I gave it in brief. And finally, we have our host. I am rewinding to that. This host is now called, what is the name of this host? Transformant. Why it is called transformant? A transformant or recombinant is the host that bears rDNA. Now, this transformant should be provided all the necessary conditions for it to multiply. So, for it to multiply, the host to multiply, then our concern is not the gene of interest, it is the protein of interest. Okay. See, this is the gene. Suppose the green color is our gene of interest. The ultimate aim of biotechnology is that gene of interest should be expressed into the protein of interest. So, this is a symbolic representation of central dogma or the stages. The gene of interest will undergo transcription to form the mRNA, and that undergoes translation to get the protein of interest. So, ultimate aim is not gene of interest, to get the protein of interest. So, gene of interest, we need the protein of interest. So, to get the protein of interest, then second, to make this multiply, it should get multiplied. To multiply the host, and not only that, for large scale production, we have to go for bioreactors. So, the next slide depicts the bioreactors. So, the advantage of bioreactors, one is for the expression of the gene of interest. As I told you, we are not concerned with the gene of interest, it should be expressed into the protein of interest. Then, second, for the reproduction of the host. We know the host is E. coli. And lastly, large scale production. The best option is bioreactors. So, the purpose of bioreactors is given here. One, for the expression of the gene of interest, because the aim is not for gene of interest, the gene of interest should get transcribed and translated to get the protein of interest. So, for the conditions should be provided for the expression of the gene of interest into the protein of interest, one. Then two, the host should multiply. So, all the optimal conditions should be provided. And finally, large scale, it should be on large scale, huge tons of this host should be multiplied in a very short duration of time, we make use of bioreactors. So these bioreactors provide optimal conditions of pH, temperature, oxygen, culture, substrates, salt, vitamins, etc. So that's the advantage. It provides all the optimal conditions favoring these three parameters: expression of the gene of interest, large scale production and reproduction of the host in minutes. Okay, so that is the advantage of using a bioreactor over other smaller vessels in the biotech lab. So we make, see the huge bioreactor. Okay, and all bioreactors have a temperature control system, pH control system, oxygen control system, sampling ports, etc. So that's inbuilt mechanism, and they most of all it has a capacity to hold 100 to 1000 liters of the culture medium. We have two types of bioreactors. The simple stirred bioreactor, A is depicting the simple stirred bioreactor and B is depicting the sparged stirred bioreactor. So both bioreactors serves the same purpose: large scale production. Second, facilitates or gives the optimal conditions for the multiplication of the host E. coli, and lastly also provides appropriate condition for the expression of the gene of interest. Okay, now we are going to analyze only the simple stirred bioreactor. It consists, see, look at the base. The base is curved. So if the base is curved, it allows mixing, even mixing of the content. That's why in our mixie jar, the base is not flat, it is curved, so that there's even mixing. Then also there's a stirrer. The purpose of the stirrer, it agitates, it stirs, and there's even stirring and mixing and also even distribution of oxygen. Then here you can see at intervals alternatively air is pumped. Okay, so that's the setup. Not only that, there's a foam control because of this agitation, continuous agitation with the stirrer and this alternating pumping of oxygen. There's chances of foam to be formed, okay, like the cola and the aerated foam to be formed. So there's a foam control system. There's a pH control system, temperature control system, and most of all there's a sampling port. Sampling port is a small tap. It's behind and not shown there. It's a small tap in between. At intervals the sample can be tested. It can be checked at periodic intervals, the stage at which it has reached. Okay, so that's called the sampling port. So once again talking about a simple stirred bioreactor, any bioreactor has a capacity to hold 100 to 1000 liters of the culture medium. Besides that, the stirred bioreactor, it has a stirrer. The purpose of the stirrer is to agitate the content, and the agitation goes on continuously, so that's even mixing and distribution of oxygen. Also look at the base. The base is curved, so this also facilitates this mixing. Then air is also pumped alternatively, and because of all the setup, what happens is chance of froth or foam to be formed on top. So to control the foam, there's a foam control system, there's a pH control system, temperature control system, and above all there's a sampling pot where samples are checked at periodic intervals to see whether the biosynthetic pathway is completed or not, because here the gene of interest is also going to be expressed. So transcription is going to occur, translation is going to occur, and we are going to get proteins. So from the sampling pots in between, we are checking whether the proteins, say insulin, is formed. Okay, so the final end product should be obtained. So that's why there's a sampling pot to check. Now we are moving on to the last slide. Very, very important after the biosynthetic pathway. So all what we dealt now is the biosynthetic pathway, the product. Say the gene of interest, say insulin gene coded for the protein insulin. Finally, the product has to undergo a series of processing. The first step is it should be separated and purified. So that is done. It's taken from the plant or it's taken from the bioreactor, separated and purified. The insulin should be purified. The next step, it is formulated with preservatives. Look, so this is the first, this is the second step where it is formulated with preservative. After formulating with preservative, it is clinically tried on some animals. See, this is the third step. Usually mice and monkeys are used for testing, clinical testing. Then the fourth step we have is the quality control. It's tested for its quality control testing, and finally it's bottled and available as a final product in the market. See, this is the final where you have it in bottle form. So the steps are given in sequence in the center. I am highlighting it. First step is separation and purification of the product. Second step is formulation with preservative. Third is clinical testing. Fourth, quality control testing, and finally it is available as a finished product in the market. See the stage. Okay, after a series of processes, the product is available. See, humulin is the insulin produced by rDNA technology, available in the market. Clear? And in this context, I will make a reference with the present scenario. We are into the third phase of the human clinical trial of COVID-19, developed by the Oxford University and manufactured by the Serum Institute of India, and now it is happening. Currently, the third phase of clinical trial on human beings is done in the Sassoon General Hospital in Maharashtra. Okay, so they are administered, and they are different persons of different stages, of different areas, and also of different situations, health conditions have volunteered. Thousands of them have volunteered to take this, and then the performance or the reactions are studied, and if it's found to be successful, it will be available shortly. Okay. So finally, only in the third phase only it is available to the human beings. Before that, there was second phase tried on human beings. The first phase was tried on animals. And in the next chapter, we are going to see how animals are used for testing of drugs as well as injections, etc., vaccines. I hope you have understood the steps of rDNA technology. Thank you.